Good morning, everybody. Good morning from Israel. Good day from the rest of the world to the rest of the world. My name is Miri Ben Chaim. I'm the director of the Division for International Studies of the Faculty of Agriculture, Food and Environment of the Hebrew University of Jerusalem. It gives me a great pleasure to open the second webinar in plant sciences under the auspices of PEARS Foundation of England. We invite everybody in the world, in Israel, in the faculty, wherever you are, to join us, and we believe that it will be extremely interesting and beneficial to all of you. There are going to be 15 seminars that are going to be presented. Out of 17 students that finished this year, the yearly program, 2 of the works are still confidential with the results, therefore they are not going to be presented, and 15 are going to be presented. We have, like I said, 17 students from Ethiopia, Nepal, Chile, India, Nigeria, Serbia, USA, Mexico, and Kenya. Enjoy the 2 days that are coming ahead. Enjoy your seminars. Feel free to join in to ask all the questions that you want. I hope you're watching in the rest of the world. And good luck to you, students. And now I would like to invite the academic presenter of this program, the head of this program, Professor Yoshua Shuki Saranga. Please.
Hello, good morning, everybody, wherever you are out there around the globe. I'm very happy to open here this second webinar on plant sciences. It's actually the peak moment for our international students that spent here a whole year of very intensive studying and conducting a research exercise, of which results will be presented here today and tomorrow. I hope you will join us, if not for the entire 2 days, then for selected lectures. And I would like to open up with the first lecture. The first is our guest lecturer, Professor Baruch Rubin from the Faculty of Agriculture in Rehovot. Baruch was the founder of this international program some 20 years ago? No.
Maybe less.
Maybe less. But anyway, he was the founder. He structured the program from the beginning and was the first academic head of the program. And Baruch will give us today a lecture on herbicide-resistant crops, GM, and herbicide-resistant weeds, cause and effect. Baruch, please.
Good morning, everybody. Needless to say how proud I am to see the 17 graduates standing or sitting here and looking for their time to present their results. When we started, it was much less students and we had less scholarship, but now, like a teenager, you are grown up. The whole program is now mature, and I hope that the the entire 2 days will be nice and contributing to your future. What I'm going to present today is the result of many years of work in our faculty and in other places in the world. And I know that you had a course on weed science, and what I'm going to present today is part of the contribution of my students, because I don't work. I work only with my finger. Do that, do that, do that, you see? And they do, and they think, and they come up with results. So let's go. I put a question mark on the cause and effect. Is this the only cause for herbicide resistance? You know that GM crops now are very common in several countries in the world, and they are going to be probably adopted by other countries in the future. So let's start with the what is a weed? I hope that everyone knows it. We call it any plant that grows out of place and causes damage. If it doesn't cause damage, it's not a weed. It's probably a wild plant or something, but it is not a weed. Weed has a connotation of damage, of injury. And in some crops, we can reach up to 80% yield losses, and the reason is competition between the weed and the crop, and they compete on water, nutrient, light, and space, and they are threatening crop health, and they are threatening also the public health. As I'll show you with one invasive weed, you can see it here. It's Ambrosia convertiflora, which is an invasive weed to Israel, invaded about 15 years ago, and now it is causing huge damage. But you can see here the flowers, when it flowers, the pollen grains are dispersed by wind and cause allergy. Allergy to people. 35% of the population in the world is sensitive to this pollen grain, and they become ill. And there are millions of working days that are lost because of the allergy. You can see it is on the Alexander River in Emek Hefer in Israel, and it is dispersed all over. You can see here in the map the red points are places where the weed is present, compared to the green points that were in 2004. The dispersal is amazing. It is a huge area covered now with this weed that causes damage to crops and also to human beings. Herbicides are one of the means that we are fighting against weeds. We can do that by human being, but this is not the way that we would like to spend our time. But herbicide is a chemical that's cultured biological organism that's used to kill or suppress the growth of plants. And we have 220 different chemicals now that are classified as herbicides. There are more than 20 herbicide mode of actions. That's the way they kill the plant. And the last mode of action has appeared on the late 1980, practically it is 1988. Since then, we have no new mode of action among the new herbicides developed. This means that we are using the same mode of action all the time, and this has a connotation that I will talk to you about later on. Look at the global pesticide market. This is the prices, $57.5 billion. This is the price that the consumer, the farmer, pays for this pesticide. You can see that 24 billions are going for herbicides, while for fungicides only 15 billion and 14 billion for insecticide, and some other products take 4 or 5 billion. But the major, almost 50% or 40% of the total market for pesticide goes for herbicide. That means herbicides are very heavy part of weight on the farmer's expenses. You can see that we are talking about 690 million hectares treated with herbicide. This is huge, given the fact that only part of the globe is agricultural land. And we are talking about a huge area. So the benefits of the chemical weed control, I don't have to elaborate on that much, but it reduces the cost of food, and the farmers, the quality is increased. There's no doubt about it. You can argue that they are toxic and they are cause damage to the environment and so on. But there are some facts that we know that we cannot run a modern agriculture everywhere in the world without using herbicide, but we have to use it in a severe or in a rational way. But there is they reduce the weed control cost, and they also enable a reduction of tillage. Tillage is one of the most expensive activities that the farmer has to run. It's in cost. It costs in fuel and in CO2 enrichment of the environment and so on. And if you can reduce it, and this is the trend in the world, to move toward minimum tillage or zero tillage, this will not be possible without herbicides. And save the CO2 emission, and above all, eliminate the need of minorities, women, and child labor, because everywhere you go in the world, and I travel the world, and I saw always the minorities are those that pull weeds in the field. No one from the rich, you can see this, I've taken it in Peru. There's 12 women with their babies on the back are pulling weeds in onion. When I told the farmer that I can solve this problem with no time, with herbicide, he said, don't do that, because if not, these ladies will not have money to survive. But this is, I would say, nonsense, because these women can go to school and learn and do much more productive work, not pulling weeds. And it's always women and children or minorities. And I will show you, this is in Madagascar. This is a rice field. And you can see this is absolutely not necessary, because we have now the means and the ways how to control this weed without these people work. This was taken in Israel. Who is working here? Arab women. That means the minorities, and within the minorities, we take the weakest part of the society and put it in pulling weeds, instead of using much more effective and cost-effective way to control weeds. This is I've taken in California. You can see, again, who are the minorities. The minorities are those who came from Mexico or from other Central America countries. This is in Canada. The Indians, immigrants, are doing the work. Again, we are talking about minorities, and this could be done excellently. This is strawberry in Ontario. And we developed here a method how to control weeds in strawberries without any involvement of manpower in hand weeding. So why these people can do something much more productive than pulling weeds in the field. However, the farmers, many times, unfortunately too many times, are using the herbicide wrongly or not according to the directions. So we have sometimes soil and water contamination. We may damage non-target. We spray in one place and some other plants will be damaged in other place, which we did not mean to do that, but because misuse or misapplication does it. We have a potential of carryover for the next crop if we are not using it according to the directions. We can shift the structure of the weed population. The weed population, I'm sure that you know that in any normal field, when you go in and you look at the society, the plant society, the structure of the weed population will be probably something like between 10 and 20 weed species will be present with the crop. This is normal, and for this we are aiming our weed management practice. But if we are using the same herbicide or too much herbicide or repeated use of the same herbicide, we are shifting the structure of the population and we are giving advantage to one or to two species. For example, you can see it here. What we have done here in this cereal, this is wheat. What you see here down in the light colored, this is wheat field. But you can see the wild oat has been shifted, the entire wheat population has been shifted toward those who are tall, and there we cannot control with herbicide. Now we have the ways how to control the weeds, but you can see that these are tall and the wheat is short, and the competition is huge. Or you can see here this is Orobanche in, you know Orobanche? It is a parasite, and this infesting carrot field up the north in Israel, and you can see this is made by man. We drove the field to this situation by our activities. We wrongly done that, and now we are fighting how to solve the problem, of course. And we have some ways how to solve it, but this shows you the shift of the wheat population because of our activities, of the farmers' activity. So, herbicide resistance. We have, the last time we are talking about 400 weeds that evolved resistance to herbicides, 217 species, and this is in 21 out of 25 herbicide side of action, mode of actions. That means all herbicides that we are using, we have weeds that evolved resistance against. That means we cannot escape from this situation. And we are talking about 148 different herbicides, and it has happened in 65 crops in 61 countries. That means we are talking about a global problem. You can go starting from Kenya to South Africa, to North America, to South America, to Middle East. Everywhere you go, you find herbicide-resistant weeds. And the evolution of these weeds is man-made. It's happened because of our misuse of herbicide. It was not a necessary or something that inevitable process. This happened because we misused and abused the herbicide treatments. So the turning point, the whole world knew about herbicide resistance, but all of a sudden it became an economic problem when the transgenic crops appeared in the market, on the market. You know what is transgenic. I don't have to elaborate on that. When you take a gene or a trait from one species and transfer it to another species in a way that couldn't be happened by normal crossing, this is transgenic. And we have several crops that now you can see here. We are talking about in 2012. This is the last available information. We are talking about something like 400 million acres, or you can multiply it by divided by 2.5. We are talking about 100 and something billion hectares of transgenic crops. Now this is, you can see, this was the developing world, the industrial world, and this is the developing world. But you can see that nowadays we are talking about half and half. The distribution is all over the world, particularly after China and India adopted the transgenic crops, and it competes with the, you can see here that the herbicide tolerant crops are the majority, more than 80%. And mostly glyphosate resistance, they introduce a gene, a trait taken from soil bacteria, and they transferred it to the plant, and it is expressed in the chloroplast because the glyphosate resistance has to be in the chloroplast. They moved everything to the gene, the transgene was moved into the chloroplast, and now we have plants that you can spray with glyphosate, as you may know as Roundup, and nothing happens, and the weeds will die, and the crop will survive. You can see that the majority are glyphosate resistance, and we have also insect resistance, BT. You see the red one, the red line, but we have also stacked both glyphosate resistance or herbicide resistance and BT or insect resistance. We have only these traits. Why we don't have yet the transgenic crops resistant to drought? Why we don't have yet crops resistant to fungi, to pathogens? It takes time. The public does not accept the transgenic crops as it is. There are some resistance. In Europe, for example, they don't like it at all. They don't approve it. So it is not in Europe. And in other countries, for example, Israel is non-GM crop, GM country. So we can divide the world into GM countries and non-GM countries. And Israel is among the non-GM. For my taste, it's wrong, but this is a decision. It is a political decision, and because we export much of our produces to Europe, and Europe may ban this export if we will go. But you can see that there are 4 major crops which are resistant to herbicides: soybean, maize, cotton, and canola. One may ask the questions, why no more crops? Why not wheat? Why not rice? You know, everyone knows about the golden rice that introduced the carotenoid producing pathway into the rice. So people who are eating only rice, like in various countries in the Southeast Asia, they can profit from that, benefit from that. And, but it is not adopted. People are maybe afraid from Frankenstein or something else. There are several ethical and maybe prejudices that inhibit the adoption of more crops. But there is no doubt this will come with time, because it has a lot of benefit, although there are some basis for concern about adopting GM crops. But this is now, it's mostly there. For example, all the papaya that Hawaii produces is the biggest producer in the world of papaya. All is transgenic, and it is not herbicide resistant but virus resistant. So, but this is a different story, and we will look at this, that the transgenic revolution, after introduction of the GM crop in 1966, most of the species are Roundup or glyphosate resistance, what we call Roundup Ready. The management practice is no tillage. That means we are not plowing or converting the soil. We plant directly into the previous crop remaining, and this is called zero tillage. I'm sure that you learned about it, and it is very important in terms of soil conservation and so on. This means that zero tillage and the presence of the transgenic crop, we are reliable too much on glyphosate, on what we call Roundup, or products that contain glyphosate. And this ended up with very fast evolution of glyphosate or Roundup resistant weeds. We are talking about 24 species now in the world. But look at this ad. This was published 2 years ago in the Delta Farm Press. And the farmer, look at the shape of the guy. He is very experienced, right? Very experienced farmer. You can see he is wearing a hat, and it's, we call it in Hebrew, balabait. You can see this is a very, very impressive person, and he says that if we pay only $3 per acre, why should I use another herbicide? This is so cheap. So what this invites you to do is to apply more and more, and again, the same herbicide, because it's so cheap. There is no cheaper herbicide for you, and total vegetation control. It controls, you know, glyphosate. It controls almost all the weeds, so, and the crop does not damage. So, oh, it's excellent solution. Why don't use it? Use it more and more and more. And the outcome of this was the 24 weed species that are resistant to glyphosate. So this selection pressure, when you are using the same herbicide more and more, and again, you are exerting a huge selection pressure on the weed population, and you end up with a big number or large number of herbicide resistant weeds. I just want to draw your attention to the lack of rotation. You know, we have crop rotation. You know what is crop rotation. But when we plant an orchard, there is no crop rotation. We plant orchard for 30, 40, 50 years. And if it is olives, we can see olives of 100 years or maybe more. So this will be monoculture because we want only the olives to grow in the field. So it's monoculture. In crops, we are talking about planting annual crop every year on the same field, like cotton, 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 cotton. And this is possible when the crop can afford it. In many crops, you cannot do that. For example, if you plant tomatoes, you cannot replant the same field with tomatoes because of soil-borne diseases and so on. But in cotton, there is no problem. In wheat, for example, in the Negev, you are using wheat fallow. That means a 3 years crop rotation, which contains only wheat, because this is the only crop that we can grow in the Negev with this amount of water available. So we are going from crop rotation to monoculture, and this is disaster in terms of because you are using the same herbicide more and more and again. So, and we have no mode of action to rotate, and we ended up with glyphosate-resistant weeds, what we call GR, glyphosate-resistant, and it was first found in Australia in Lolium rigidum, and now we are talking about very, very much, very devastating weeds. Look at this. It's Amaranthus palmeri in cotton in Georgia, and this zero yield. No yield can be harvested from this field. It's the same thing. You can see they found here that the resistance to glyphosate is due to gene amplification. They found that the gene responsible for the target site for glyphosate, the EPSP synthase, was amplified from 10 to 150 times in the genome of this weed. That means even if you applied 2 times the rate, recommended rate, you have so many copies of the EPSP synthase that can continue and produce the aromatic amino acids with no limitation. So this is very unique mode of resistance, mechanism of resistance, that gene amplification. Last week, we just found a paper. This is the paper that published in PNAS, in PNAS, describing the resistance mechanism. And you can see it became superweed. We call it superweed. And this is in America, Scientific American in 2011, a couple of years ago. And you see the pathway for resistance is here. Now, the new or very close species, Amaranthus tuberculatus or Amaranthus rudis, are synonyms, and also develop glyphosate resistant, but due to different mechanisms, alter target sites and non-target sites, resistance mechanism. And what happened now that this superweed, what nearly half of all US farms now have superweeds, which means that we are talking about a problem not in one farm in a region, but we are talking about more than half of the farms in the United States. That means that we are in real troubles. Now this is soybean in Argentina. There is any Argentinian here? No. But we are talking about 4 million hectares in 2013 infested with this Johnson grass or Sorghum alepense. In Hebrew, we call it kusab. You know this weed? No. But this weed, look at this here. This weed in soybean treated with 9 liters of Roundup, when the recommended rate is 2 liters. That means this was sprayed with 4.5 times more herbicide and nothing happened. The same thing with 6 liters. So the problem is devastating in Argentina. This I've taken in Australia, Lolium rigidum, resistant to glyphosate in Australia. And so glyphosate resistance evolved not only in the United States, but also in Europe, Spain, France, Greece, Portugal, Italy, Poland, Czech Republic. But remember that in Europe, there is no transgenic crops. That means you don't need the GM crop in order to get glyphosate resistant. But this happened in orchards, in olive orchards, in citrus orchards, in vineyards. When you spray glyphosate 2, 3, 4, 5, 6 times a year and exerts the selection pressure not by residual herbicide, by the repeated use of the same herbicide once and again. So this is also in Asia Pacific, in Australia, China, Israel, Japan, Malaysia, New Zealand. So it is not only in the United States where they are using. Now, I put red color on the countries where transgenic crops are grown. But China does not grow Roundup Ready crops because they have manpower to handle it. And they adopted the BT, like in India and in China. They did not adopt the Roundup Ready. They adopted the BT, the insect resistant. Now in South and Central America, Argentina, Brazil, Chile, Colombia, Mexico, Paraguay. In Argentina, I told you also only the 4 million hectares. It's for you to know, it's 40 million dunam. It's about 40 times the cultivated land in Israel, the entire cultivated land, irrigated. So we are talking about South Africa. South Africa has adopted cotton, Roundup Ready cotton, so it might be associated with GM. But in all these other countries, particularly in Europe, the problem is not the transgenic, but the misuse. This is the report from Japan on glyphosate-resistant Tanya ryegrass. Japan is very strict about transgenic crops. They do not want to adopt, in fact, but they, because of misuse of herbicide, they have resistance. Now this is in Israel, not very far from here, in Kvuzat Chile. When you go south, you just cross it. You see, this is Coniza bonariensis, resistant to glyphosate. You can see it here. There are 2, and those of you who know Ziv Kleinman is doing his PhD on that. We have glyphosate resistance whenever there is a selection pressure. If we press and put pressure on the weed population, we end up with resistance, no matter what we do against it, unless we do it in a way that the selection pressure is changing every season or every year by using different mode of action. So repeated application, it doesn't have to be GM crops. You can do it in orchard, in roadsides, and fallows. We are using a high rate because the herbicide is cheap. is cheaper per dunam than a can of Coca-Cola. You can, for a can of Coca-Cola, 5 shekel, you can treat 2 dunams. So this means it's very cheap. So why avoid using it? No tillage, which is very important from the soil conservation, but it is a problem with the herbicide resistant. All the research and work that we are doing in the world, including in Australia, does not help. You see, this is resistant to dichlorphop in 1999. It was 46% of the fields. Now it's 96% of the fields. That means all the work and what we are doing in the lab and in the field does not help, because the farmers are going and misusing the herbicide and more. And now it's virtually all fields in Western Australia are resistant to dichlorphop. And the same thing happens with sulfometron or with ALS inhibitors. From 65% in 1999 to virtually all the fields contain weeds which are resistant to this group of herbicide. So what's the solution? The solution is to introduce new herbicide-resistant crops. That's what the industry are doing. And just, I will not elaborate all this, the new going to be introduced. This one, last week, received approval from the United States with resistance to HPPD. That means instead of stopping, hey, wait, let us think what we can do against this horrible problem. And before we adopt a new trace. So that's what they do. And I conclude my talk with several statements. First of all, we cannot run a modern agriculture without herbicide. Remember that. This is all the organic approach is nice for small level agriculture, for a smallholder, for, I would call it gardens. In fact, 2 to 5% of the whole agriculture in the world is organic. We cannot convert the whole agriculture into organic. This is not practical. We cannot feed the starving people in the world with organic food. Forget it. So we have to use herbicide, but we have to use it in a way that takes into consideration all the outcomes. And first of all, if we will continue as we are doing now, we are running into troubles. There is no new mode of action, so we have to look for a new mode of action. The new generation of herbicide resistance will come up with multiple resistance, and this will be disaster. We have to change the regulation. I think that we have to change the regulation and apply much stricter penalties on those who are using herbicide without obeying the label and the recommendation. Because if resistance will continue to evolve in the current pace, the benefit of chemical weed control will be nil. Now, just because you were so nice and so kind, I'll give you a brief introduction to what happened with herbicide resistance in Israel. This is just a very short knowledge. We have 28 cases of herbicide resistance in Israel already. For a small country, we are probably after Australia, the highest number of herbicide resistance per acre in the world, per dunam or per hectare. This is Brachypodium, already in 1973, and this is not chrysanthemum field. This is a wheat field. We have to have grains in here, but we don't. We have a nice lady but nice flowers, but the farmers cannot make anything out of this. This is a wheat field in the Negev, which is infested with herbicide-resistant chrysanthemum. You can see this field. We have run some experiment there, and you can see we can solve the problem by good selection and rational selection of herbicide. We can solve the problem. Here, there is no chrysanthemum. Here, this one was treated with tribenuron. Look at this. It's a carpet. It's a beautiful picture, but the farmer has to survive on this field, not on the chrysanthemum. This is cotton field not very far from here in Kibbutz Na'an. You can see this is Amaranthus palmeri that develop resistance to herbicides. This is Phalaris minor from Gilat. You have been to Gilat in the Negev. This was taken, look at the sensitive one against the resistant one. And this is, again, wheat field, but it's only Phalaris paradoxa, not very far, in Revadim. We know exactly the mechanism of resistance, including the molecular, and the same thing with wild oat resistance to herbicide. Lolium rigidum, multiple herbicide resistant. And with this, I would like to thank you very much and thank my students and my colleagues that were part of this study. Thank you very much. Question?
As always, Baruch is a fascinating speaker, so I couldn't stop him at the time. So we are running behind schedule, so we can have time maybe for 1 or 2 quick questions, please, and quick answers.
Quick answers, thank you.
All right.
Good.
Thank you very much, Baruch.
Thank you.
And I would like to move on to the students' presentations. And the first one, Javiera Aravena-Calvo from Chile. Please, Javiera.
Hi, good morning. My name is Javiera. I am from Chile, and I'm going to present you my research exercise called Identification of Genes Involved in Scent Regulation in Petunia Flowers. My research was supervised by Professor Alexander Weinstein. Well, what is flower scent? Flower scent is a mixture of volatile molecules that are produced by flowers. The flower scent is very important in order to attract pollinators to assure the survival of the next generation of the species. Also, scent production is very important for humans, mainly in agriculture, because in order to have a good yield in the crop, we need a good pollination. So the crop must be successful to attract them. Also, flower scent is widely used in the perfume industry, also pharmaceutical and food industry. Well, a particular blend of different volatile molecules and the level of them will determine the scent we perceive in particular flowers. Thousands of volatiles has been described, and they belong to three main families. The first one are the terpenes, as we see here, the linalool or limonene that make the special scent of the orange blossom. Another family are the benzenoids that are produced by cinnamon, roses, almonds, and also petunia flowers. And the last group is fatty acid derivatives, but there are also other molecules with a miscellaneous configuration. Well, in this research, I focus in the phenylpropanoids and benzenoids compounds, and its biosynthesis start in Petunia flower, start from phosphoenolpyruvate and erythrose phosphate that enters to the shikimate pathway. The shikimate undergoes several reactions to yield phenylalanine. Then phenylalanine, it's going to different branches of the phenylpropanoid pathway in order to yield the different volatiles that are produced in Petunia flowers. Also, in some branches, there is a branch that also produces the anthocyanin that gives the particular color to the Petunia. Flower scent is tightly regulated for by many factors. Flower scent is regulated spatially. That means in different structure in the flower, different volatiles are produced. For example, in the Petunia, the volatiles are produced only in the petals, but not in the tube and in other structures. Also, daytime has a strong effect on flower scent. As we see here in Petunia flower, the peak of emission of volatiles is during the evening. That coincides with the nocturnal behavior of the pollinator, that is moth, in the case of Petunia. Also, the developmental status plays a role, as we see here in this graph. During the first stages of development of the flower, there is almost no volatiles produced, but when the flowers opens, the volatiles start to be produced with the peak of emission after 1 to 2 days after anthesis. Well, and also the genetic background and environmental factors has an effect. What about the regulation of flower scent? Up to now, 4 transcription factors has been described in the literature to regulate the biosynthesis of the phenylpropanoids and benthanoids. The first one is odorant one that is described to regulate mainly the enzymes of the shikimate pathway. The other one is EOB2 that it regulates indirectly the shikimate pathway through odorant 1 and regulates directly the phenylpropanoid pathway. Recently in our lab was discovered EOB1 that regulates by itself the phenylpropanoid pathway, but also was described that is having a negative feedback, a relation with odorant 1. So it's also controlling indirectly the shikimate pathway. And both of them are downstream the main transcription factor that is EOB2. The last one is a repressor of this particular enzyme. So this repressor will determine the synthesis of particularly isogenol and eugenol. So as we can see, these transcription factors are regulating this pathway, and the behavior of them will determine the substrate availability and the different fates of each compound. Now we know more or less how is regulated floral scent at molecular level, but we still don't know how the transcription factors are activated to regulate the scent production during the development of the flower. So what is switching on the biosynthesis? So we think that maybe hormones are playing a role during the development of the flowers. We think maybe the jasminic acid can be involved because it is known that its main role in plant is to regulate secondary metabolism, as for example anthocyanins and nicotine. Also, it is involved in herbivory and defense against pathogens with interaction with salicylic acid and ethylene. And also, it is present in different steps of plant development, as a flower or senescence and tuber formation. Now the signaling pathway of jasminic acid, I show here in this figure. When in the resting state in the plant, there is a low concentration of active jasminic acid. So the basal state is that just protein, together with other core repressors, are repressing the transcription factor MYC. Under stimulation, for example, herbivory, the levels of jasminic acid go up, and just interacts with CoE1 and the F-box complex mediating jasminic acid, and all this complex is destroying the repressor. So all this complex is released, and the transcription factor can activate the promoters of the jasminic acid responsive genes. Among these genes are all the genes of the biosynthesis of jasminic acid, and also the repressor JAS and the MYC transcription factor. So we can say that this this signaling pathway is self-regulated. The aim of my research was to determine if genes participating in jasminic signaling are involved in regulation of floral scent. If we come back to the diagram of the signaling pathway, we see that the three main regulators of this signaling are the JAS repressor, the transcription factor MYC, and CoE1. In my research, I focus only in JAS, the repressor. Now, how to see if this gene is involved in flower scent production? Our approach was to use virus-induced gene silencing, or VIGS, that is based mainly in the plant defense through interference RNA. How it works? When the plant is infected with a virus, the plant will develop a defense mechanism against the virus. And the way to destroy the virus is silencing all the viral genes. So if we put between the viral gene a gene of interest, this gene also will be silenced, and we will see a suppression of the expression of the gene. This technique is widely used in different species. It's a very good approach because it doesn't cause any damage during the development of the plant. But the disadvantage is that it's not uniformly dispersed, the virus. So in our laboratory for Petunia, inside the virus genome, we put the chalcone synthase gene. That is the gene that participates in the biosynthesis of anthocyanin that gives the purple color to the wild type Petunia. As we put it here, inside the virus, the plants infected will show no anthocyanins production, so they will look white. So this gene is just a reporter gene to see where the virus is active. So together with this gene, we put our gene of interest, in my case, JAS gene, to see if how it is silenced in the Petunia. So schematically, the methodology was developing VIGS. For that, I needed to clone the gene that I look in the cDNA library, and then I infected the plants. And after the plants showed the phenotype, I analyzed gene expression by real-time PCR, and I did volatiles analysis by headspace and pool extraction. The cloning, well, I went to the Petunia 454 cDNA library available in my lab, and I found a sequence called isotic 06320 that has high similarity to other just genes described in the literature. So I took this sequence, I cloned it in pGMT-EC in order to amplify the sequence. Then I cut it with restriction enzymes. I put it in the final vector, PTRB2 containing the CHS. And then I agroinoculated Petunia plantlets. the sequence I found, the isotic 06320, was very similar with other gases from tobacco, Catharanthus roseus, and tomato. As we see here, the two particular domains, characteristic domains of these proteins are the TIFI domain, that is the responsible for the interaction with the core repressors, and the JAS motif, that is the responsible of the interaction with the transcription factors that are repressed. Well, around 1 month after the infection, I saw the phenotype. I got white flowers with some purple patches, but compared to the wild type, they look very good. So then I took white flowers and I did RNA extraction to analyze gene expression by qPCR. The result of this qPCR was that I was really suppressing the expression of this gene. As we see here, the gene was suppressed almost 3 times compared to the control plants. And interestingly, also I saw a decrease in the expression of ODRAN1 that I mentioned before. It's a transcription factor that is regulating the shikimate pathway of the volatiles. About volatiles analysis, I did 2 analyses. One is the dynamic headspace. That is the analysis of all the volatiles that are released by the flower. In order to do this, I put the flowers for 24 hours in hermetically sealed flasks, and all the volatiles produced were trapped in columns, and then this column was eluted with hexane, and then the volatiles were analyzed by gas chromatography, mass spectrometry. The other analysis I did was the internal pool. That is the volatiles that are in the petals that are not yet volatile. So I extracted them with hexane, and also they went to GCMS. Here I show the result of the pool, the internal pool volatiles, because I'm still working with the headspace. The headspace is giving, normally it gives very variable results, so I need to do many replicates. So I did many, but I'm still waiting for the analysis of the chromatography. So here I show the internal pools, and as we can see, there is a slight tendency for benzyl alcohol, picrosol, eugenol, isogenol, and benzyl benzoate. They were decreased in the silenced plants, although most of the volatiles remained constant, but we can see some effect of the suppression of this gene. So as a conclusion, according to the expression, gene expression results and the volatiles, we can say that probably JAS is needed for the ODRAN1 expression, because it was, the expression went down when JAS was suppressed. So it would mean that maybe jasmonic acid may play a negative role in scent, because when jasmonic acid is present, JAS protein is destroyed. So it's the same effect as we are suppressing the gene. So, but still further experiments are needed in order to demonstrate if jasmonic acid is having or not an effect on flower scent production. So my future work will be to characterize other JAS proteins, because in the literature, for example, in Arabidopsis, there are 12, and all of them has different function. In tobacco, there are 10. I only focus on one of them. So it will be interesting to see what happened with other copies of this, with other genes, and also with the other genes of the pathway, as COE1 or the MYC transcription factor. Also, it would be interesting to see if JAS protein is interacting with the transcription factors that are regulating the phenylpropanoid pathway. And also, we will study the direct effect of jasminic acid application in flower scent production in Petunia flowers. So I want to thanks to my mentor for receiving me in the lab and for guiding me during this research. I want to thanks to the people of my lab and also to PEARS Foundation to allow us to present our research here. Thank you for your attention.
Thank you, Javiera. We are ready for questions. There will be. Yes, Ranjit, please.
I have one question for you actually. In one slide you showed that there is increase in aromatic compounds in plants after the flowering.
Here?
Yeah. My question in general, I don't know if it's related to your subject or not, but the FT genes are responsible for the increasing flowering of the plants.
Sorry, again. The
FT genes.
FT.
In
Okay.
most of plants, they are responsible for the flowering.
Uh-huh.
Will they affect the essential oil production of the plant?
FT. No, no. Actually, I don't know. I haven't read anything about FT and flower scent production. But I know, yeah, they are involved in the development of the flower. But no, I didn't find anything about FT. Yeah.
You mentioned something about EOB1, right?
Yes.
Yeah, you said the ODO1 negates the EOB1. I didn't really get what you were trying to expatiate there. You mentioned something about the propanoid pathway. The EOB1 has a negative effect to the ODO1.
Here, for example, okay. No, it's just EOB1 is a transcription factor that is controlled by EOB2, and this is having a positive effect on this transcription factor. But this transcription factor, when it's expressed, is having a negative effect on EOB1, so that there is a negative feedback only between them. That's it.
Yes.
Were you trying to know the volatiles present, or how do you do that?
The volatile for the headspace, maybe? Here? Yeah, okay, I did two analyses. One was to measuring the volatiles that the flower produce, that they are transferred to the air. So for that, I put the flowers in closed flask, and all the volatiles were trapped in a column. And then that column I eluted with Xen, so I could extract the volatiles that were volatilized by the flower. The other analysis I did was the volatiles that are being produced in the petals, that they are not volatilized yet. So, did I answer your question?
So extracting the compounds that would become volatile
Yes.
at the time.
Mm-hmm.
So what the work you've done now, what is the significance to the general public? Like, you studied jasminic acids.
Yeah,
if jasminic acid is increased or something, will it increase the volatile content, or how would we use your work now in the
okay.
open world? What is the usefulness of this
Okay,
research?
up to now, there is no research published about the involvement of hormones in regulation of flower scent. There is nothing about. So this work will contribute to know to a better knowledge if hormones are playing a role, and this is basically to to understand better the pathway, because still there are many steps and the regulation that are not well known still.
And do you know if commercially it has been tried to increase certain level of hormones or something in order to increase the chances of seeing and also to make a
commercially, I don't think so, but there are many reports about making transgenic plants, transgenic roses or Petunia that are producing different blends of volatiles and different levels of volatile. It is possible, but at commercial level, I don't think it is done now, but it is possible, yeah. Then?
I was wondering for the dynamic headspace if the release of the volatiles is temporal, as you mentioned earlier.
Yes.
So if you can just describe how you designed the experiment to optimize.
Okay, in this case, I extracted all the volatiles that were produced during the 24 hours. Here I am not looking any pattern. So it will be interesting for future work to see how the suppression of JAS or other genes or the effect of jasminic acid is having in different date times. So that will be the future work, because it would be interesting to see how, because the volatiles are produced mainly during the night. So it would be nice to see if during the night there is a change in the dynamic. Mm-hmm.
Can I elaborate a little bit more on the question of Olabi? Once you learn the hormonal control on volatiles, could there be any way like to manipulate the plants or the timing of the volatile release so that some more useful things can be done out of this, like if you want to collect volatiles or something? Do you see any kind of such application, like a kind of switching on and off the volatile release?
It's unknown the switch in the during volatile production. So if we know what is making this switch, we can manipulate it for commercial purposes, for example. Yeah.
Okay, so please join me in thanking Javiera.